Circuit buddy, today we will be discussing about the DNA polymerase 3 holoenzyme in E. coli. DNA polymerase 3 holoenzyme is a very complicated and multimedic enzyme, but for our understanding purpose we are taking only the simplified version of the enzyme as per Musta. Here you can see that these are the alpha subunits, these are the tau subunits, these are the epsilon and theta subunits, the beta clams are present here, and these are the gamma and delta subunits. Now let us talk about the subunits in details. The alpha subunit is a DNAE gene product. Okay. Now you can remember this by this word allele. Okay. Now A means alpha and E means DNAE. The molecular weight of the subunit is 130 kilodalton, and you can easily remember this by the word Thomas scale. I am using the mnemonic methods. If you don't know about mnemonic methods, you can search the internet and you will get plenty of videos about mnemonic methods. Mnemonic methods, T means 1, M means 3 and S means 0. Okay. Now let's talk about the tau subunits. The tau subunits are DNAX gene product, and you can easily remember this by the word tax. Okay, so T means tau and X means DNA X gene. The molecular weight of tau subunits is 71 kilodalton, and you can easily remember by this word C is equal to 7 and T is equal to 1 in mnemonic methods. Okay, so you can see that these words are helping you to remember the molecular weights of alpha and tau subunits respectively. Now the function of tau subunit is the dimerization of the catalytic core of the enzyme. Okay, now let us talk about the beta subunits. Okay, so the beta subunits are DNA N gene product. Okay, now you can easily remember this by this word bangles. Now bangles are ornaments. Okay, you know. Now B means beta and N means DNA N gene. Okay, the molecular weight of these beta subunit is 40 kilodalton, and you can remember this by this very simple trick. Suppose the bangles are 40 rupees each. Okay, so 40 kilodalton. The main function of the beta subunit is to form a clamp around the DNA template strand. Okay, so it holds the DNA template like this, like a clamp, and helps the DNA polymerase holoenzyme to move or slide over the DNA template strand. Okay, and it keeps the DNA polymerase from falling off from the DNA template strand. Okay, so that's a very important function of beta subunit. Okay, now let's talk about the epsilon subunits. The epsilon subunits are DNA Q gene products. Okay, and it has a molecular weight of 25 kilodalton. You can remember the gene by this simple trick, that is exquisite. The word is exquisite. Okay, now here E means epsilon and exquisite Q means DNAQ gene. Okay, so it is a DNAQ gene product, and exquisite means beautiful or extremely beautiful. Suppose there is a very beautiful or extremely beautiful woman of 25 years. So you can easily remember this by this trick. Now the epsilon subunit is tightly bound to the alpha subunit and helps in the slightly proofreading activity. Okay, here the theta subunits are there, and let us talk about them. There is a theta subunits. So the theta subunits are It is not DNA E, it is Hol E. So Hol E gene product, okay? And you can remember this by this word thron, okay? So thron, TH means theta and E means Hol E, gene, okay? And the molecular weight of the theta subunit is 10 kilodalton, and it's very easy to remember, I think, so I'm not giving you tricks on this subunit. And the main function of the theta subunit is to bind to the epsilon subunit tightly so that the epsilon subunit can perform its activity or function well. Now let's talk about the gamma subunit and the delta subunits, okay? Now the gamma subunits are also the product of the DNA X gene product, the same gene from where the tau subunits are produced, okay? So the gamma subunits are actually DNA X gene product, and they have a molecular weight of 52 kilodalton, and the delta subunits are also associated with the gamma subunits and helping it to perform its function, okay? So that's all about it. The gamma subunits is attached to the delta subunit, and the tau along with the gamma and the delta perform the single stranded DNA dependent ATPase activity, okay? So single stranded, let me write somewhere, single stranded DNA dependent ATPase activity, okay? Now the tau, the gamma and the delta, okay? Now that's basically the most important informations about these subunits. The gamma subunit is 52 kilodalton as I have told you earlier, and the delta subunit is 32 kilodalton in molecular weight, okay? So that's all about the simplified version of the DNA polymerase 3 holoenzyme in E. coli as per Snuster. If you found the video helpful, please like, comment and share the video, and please don't forget to subscribe my channel to get more videos like this, more trick-based videos like this. Thank you very much. Have a great day.